My name is Kavya. I'm from India, Bangalore, and I'm carrying my research work from Bangalore University Central College, Department of Biochemistry. Previously, I've got an opportunity to work with carrying out next generation sequencing of small RNA, microRNA, and this was an opportunity for me to extend my knowledge on CRISPR Cas9 tool and to understand the mode of how the sequencing is carried out using CRISPR Cas9. Experience attending this workshop, and we got a chance to learn a lot about genome sequencing. Having studied and come from a background where we have worked with NGS, there were minute details which we were not familiar with, which got clarified. And there were also people who had absolutely no knowledge about NGS, and this was a very good opportunity for them to learn about it. Apart from that, we also learned about CRISPR and its application in cancer research and stuff like that, so which was quite enriching. The only regret would be that we didn't get to use, we did get to use the software developed by Mr. Yash, which is called as CRISPR, but we did not get a copy of it. So we look forward to, getting it, because it's a very helpful and very unique tool that we have come across, which makes the sequencing easier and user-friendly. And thanks a lot, and this was really an experience which was enriching and very educative, and I highly recommend this kind of workshop. Thank you.
So I'm Nadia Suni from the University of Sciences. University of Sciences I'm geneticists. And what to say about this course? First of all, I am very, very happy to attend this course for many reasons. First of all, I have this opportunity to meet you and Yash and trainers and participants. So I would just say that the course was very clear, very nice. Thank you for you and Yash. The second reason is I learned what is exactly CRISPR Cas9 and NGS. And this course gave me this also this opportunity to know that to do NGS we should have some bioinformatics. And next time we get an NGS machine at the hospital for cancer diseases, so we should get all this information how to use it for molecular diagnostics. So in my case, when I'm lecturer for genetics, I note that I have big lack on bioinformatics, so it's good. Even I have this like this I follow the course because it has been very clear and you give many chapters and explanations. So, but we should begin with bioinformatics and then NGS and how to use both technologies for molecular diagnostics. So my next course plan is to follow bioinformatics course, which is very important. Thank you very much.
My name is Roba Hamed. I'm from Egypt. I'm doing a master's degree in cancer. Okay, I have applied to this workshop, and I was really have many doubts about CRISPR and these things. But actually, this workshop helped me a lot. First of all, it gave me a good background about CRISPR and how to go with my project using it. This for CRISPR and for the NGS. Actually, I have nothing about that, and the workshop gave me not a background, actually it gave me a good base to go with it, and I'm not using it now in my project, but now I wasn't using it in my project, but now actually I'm thinking to put it inside it and use it to analyze my data more, to be more precise. This workshop only needs something that it needs to know about, to not just read about the NGS if you have no data, no background about it. They gave it the background, but to be more familiar with it, to not make some troubles for others. And thank you so much. I'm really, I had a very good time with you, and looking forward to see you again. Bye.